Responsiveness is chemical signals and electrical change. It triggers a wave of excitation. Epimysium, like the epidermis of the muscle, makes up all the muscle tendons. Perimysium encloses the fossicle, the bundle of muscle cells. Endomysium is the wrapping around the muscle fibers. Striations are the dark A and light A band. Relaxation requires energy. Sarcomere is the smallest contractile muscle fiber unit. The thin myofilaments have the hands, and the thick myofilaments get pulled on and have the actin binding sites. Myosin heads from the portion of the thick myomin filament bind to actin building sites. Tropomyosin covers troponin on that. Tropomyosin covers the actin binding sites, and troponin is what pulls them to make them move. Dystrophin links outermost thin myofilaments to the sarcolemma. Muscle dystrophy. Sarcoplasmic reticulum is the highly modified, smooth endoplasmic reticulum. Houses, stores, and reduces calcium. Myosin heads are allowed to bind to actin, forming cross bridges. Sliding filament model of contraction states that during contraction, thin filaments slide past thick filaments, causing actin and myosin to overlap more. Neither thick or thin change length. During a relaxed sarcomere, Z discs are closer together. For a muscle contraction, first its nerve stimulation, then its action potential and electric current must be generated in the sarcolemma. Then third, action potential must be propagated along the sarcolemma. And four, intracellular calcium levels must rise briefly, and when they rise, they form cross bridges. Step 1, action potential arrives at an axon. Step 2, voltage-gated calcium channels open. Step 3, calcium entry causes acetylcholine receptors to be released by exocytosis. Step 4, acetylcholine diffuses across the synaptic cleft and binds to its receptors on the sarcolemma. Step 5, acetylcholine binding opens ion channels in the receptors that allow simultaneous passage of sodium into the muscle and potassium out. Step 6, acetylcholine effects are terminated by its breakdown into synaptic cleft by acetylcholine stearase and diffusion away from the junction. When a nerve impulse reaches a neuromuscular junction, acetylcholine is released. NMJ is a synapse. For every AP, Na in, potassium out, K plus out. APs happen by diffusion. APs are action potentials. An end plate motor, step 2 of the muscle contraction, voltage-gated ions. Depolarization all becomes sodium. Repolarization becomes potassium. Potassium is in high control inside of cells. K plus leaks out equals repolarize. Action potential, depolarization means it goes up. Repolarization, and the chart goes down due to K plus exit. Na plus channels close, K channels open, and it goes to plus 30 at an action potential. Cross bridge cycle is where the first time ATP is used. ATP is needed 3 times in this whole process. Cross bridge cycling, it's needed when you pump calcium back into the sarcoplasmic reticulum, and it's also needed when you pump sodium and pump to reset ionic conditions. The first period is a latent period, the second period is the period of contraction, and the third is the period of relaxation, where pumping calcium back into the sarcoplasmic reticulum is a slow process. And that's the longest part. Different fibers take a different amount of time to use, depending on the stimulus. Trap phenomenon takes 10 to 20 stimuli per second. When we increase how much we stim, there's not enough resting period. What's that mean? Muscles typically operate in unfused tetanus, and we don't feel because the CNS switches smoothly between motor units. The size principle of recruitment. I don't know what that means. Optimum resting rate is how stretched and has to do with the size of the sarcomeres, muscle tone, and its activation by the CNS. Isometric is when the muscle is the same length with no movement but develops tension. Isotonic is where the muscle tension remains constant. Wait, so isotonic is when there's no movement, and no, isometric is when there's no movement and it's just a hold. Isotonic is when it's like a bicep curl. Glycolysis to split a glucose happens in cytosol. Anaerobic pathway. Lactic acid leads to fatigue. Myoglobin is where oxygen is stored. Muscle fatigue. ATP synthesis declines as glycolysis is consumed. The greater the load, the briefer the duration of muscle shortening. Chemical factors in the contraction of smooth muscle include hormones, high CO2, pH, and low oxygen. Small motor units and fine control are in the multi-unit smooth muscle. In the single unit smooth muscle, the large motor units are more power instead of fine control.